This is McHardy, and I'll be telling you today about CAMI and the next steps of what we have planned. So CAMI stands for Critical Assessment of Metagenome Interpretation. It's a community initiative that aims to derive the state-of-the-art for different computational methods for metagenome data analysis, and it does so by organizing community-driven benchmarking challenges. So just as a brief introduction, of course, to this audience, it's probably not that necessary, but computational metagenomics, as you know, metagenomics is a science where we start with DNA isolated directly from an environment of interest, for example, surrounding the roots of a plant, and then bypassing the more traditional way of cultivation, we directly sequence that DNA to get a lot of the sequence fragments. And these tend to be rather short, where we need assembly algorithms that puzzle them together to longer continuous stretches of DNA sequence, which are also known as contigs. Because these contigs can originate from any member in the community, there's a special class of algorithms that has been developed for the metagenomics field, which are the genome or taxonomic binning tools. And these, as their names say, group those contigs sequence fragments into bins, which are denoted here by the colors, that ideally come and reflect individual genomes, but they can also be higher level taxonomic groupings. So genome bins typically don't have a taxonomic label per se, whereas taxonomic binning methods assign a taxon ID. When we have these bins, they can, if they're more or less, they're large, they can be draft genomes of rather nice quality for some of the more abundant organisms. Then we can go and annotate these, do comparative genome, pangenome analysis, look at functions, metabolic pathways. So try to, what biologists also are interested in when studying individual isolate genomes. Then there's another class of tools, which are the so-called profiling tools, and there's functional and taxonomic profilers, whereas what's shown here is the taxonomic profiling, where the methods aim to infer, usually from the reads, the abundances and identities of the community members of the sample that we've sequenced, and for individual ranks, and these can then, the relative abundances can sum up to 1, but they can also be a considerable fraction of a sample that just remains unassigned. So not necessarily everything can be assigned at every taxonomic rank. So the metagenomics field now is already gaining some maturity, and there's like a really lot of activity in method developments. And before this benchmarking, the importance became evident about systematic benchmarking, there was really a big problem in that there was no consensus of how to assess all of these tools, on which data sets, which metrics should be used, so how and what defines a good tool, what does it have to, or what properties does it have to have. And so this made it extremely difficult for people to compare results across papers and samples. And that's why in 2014, during a stay at the Isaac Newton Institute in a computational metagenomics program, I founded, together with 2 colleagues, Alex Gilbert and Thomas Batte, the CAMI initiative. And we kickstarted the first round of challenges there with a lot of colleagues. And CAMI has the aim to create as comprehensive and objective as possible evaluation of metagenomic software. And so involving people, we've asked them what are the principles we need to make that possible. And so all the design decisions are made by the community. So how the benchmarking should be done, on which data sets, which metrics, and how the contests should be run. And then it was decided that it's really important that we have high quality data sets. They need to be extensive, so reflective of different scenarios that are commonly used in the field. And importantly, they need to include unpublished data, so genomes of organisms that are not found in reference databases. Because in a real scenario, for the data you have from a metagenome of a microbial community, that's just a situation that applies to a substantial fraction of your sample. The evaluation measures, they should be informative to developers and applied researchers, and also very important is that everything should be as reproducible as possible, so that if we do identify methods and settings that are performed particularly well for certain research questions, that we can reproduce and make use of them. And the focus of CAMI is on these common method categories that are metagenome specific, so we do not overlap with other benchmarking challenges such as CAFA or CAGI. So there's a range of related initiatives with the aim of assessing the state of the art. So in line with improving reproducibility, there's a lot of software that has come out to make the benchmarking of CAMI reproducible. So we've implemented ISMB that implements metrics and visualizations for assessing metagenome billing tools. The Opal software that implements the same for taxonomic profiling evaluation. CAMI-Tax, which is a software where we've implemented a way to assign taxon labels for microbial genomes that go into our benchmark data sets. Metagenome has been adapted, so this is a benchmarking software allowing to assess assemblies, metagenome assemblies, and this has been adapted to handle the CAMI benchmark data sets. And then CAMI-Sim is a simulator that simulates microbial communities in terms of the taxa and their abundance distributions, and then the metagenome data generated from those when sequenced with different technologies at different depths. And so the publication of the results of the second round of CAMI challenges now is already 2 years ago. And so as you can see, we focus on these common categories here that I outlined in the beginning. In addition, we had a pathogen detection challenge, where we focused on the problem of finding the etiological agents in infectious diseases, so in clinical and metagenomics, which is, I think, quite exciting application of this technology. And just to give you really a quick walk through the past challenges, we had both unpublished and public genomes. It was decided to include the public, really high quality ones to be able to assess the assemblers particularly well regarding their ability to resolve repetitive elements. We had varying numbers of samples, up to 100, because some of the billing tools perform best with those, and we include both long and short read data. We have environment-specific data sets, so reflective in their genomes and taxonomic profiles for a marine environment and a plant-associated community. And we had a really high strain diversity data set. And so in terms of data, the samples comprised between 400 gigabase pairs of sequences, so at most, and they were created from almost 1,700 microbial genomes, which included also fungi, plant hosts, and also high copy circular elements, and both public and private genomes. And we had, before the start of this challenge, more than 350 registrations of people that were interested in accessing these data that we generated. And then in the end, 30 teams managed to submit their results on time. So there was a timeline until which results could be submitted, and then afterwards evaluations began. And we had overall more than 5,000 results for 76 different programs and versions thereof. So a lot of assemblers across different data sets running on either pooled or single assemblies and using only short read or both short and long read data. Then a lot of different genome binners, taxonomic binners, and we assessed whether they're running better on reads or perfect so-called gold standard assemblies. A lot of taxonomic profiling tools run across all the different data sets, and the clinical metagenomics pathogen detection tools. And so I'm not going to walk you through the results of CAMI, but now we'll come to discussing the next round of CAMI challenges that we're currently in the process of putting together, and where we would love to have your inputs. So the idea here is that we would try to put together human microbiome challenges So because we had plant and marine, and for the human microbiome, we had metagenomics challenges, and we provided so-called toy data sets in the past, but these were made exclusively from public data, so they are not really reflective of the challenge situation, where it's more challenging because if you have a tool that is utilizing reference data for assignments, like many profilers or binners do, then it really makes a difference whether it's in your reference collection or not. And the idea is that we want to again provide metagenomic data sets. We would again provide special data sets for the pathogen detection challenges from patients with infections. And then we will also want to provide a metatranscriptome challenge, because this is something that many people have asked about. And here we're very much still in the design phase. So the human metagenome challenge, the idea would be we do multi-body sites samples, and we would focus on samples with a more, somewhat more higher biomasses, such as the gastrointestinal tract, oral tract, airway, skin, and urogenital. And probably about 10 samples per body size until somebody tells us we have to do something different. That's really important. We would again include high quality public genomes as well as novel genomes provided by partners. So whoever wants to provide data to us, the idea is that the data will be confidentially included in complex mixtures so that nobody can really just analyze your genomes by themselves. And they can only be used for the purpose of the challenges, for nothing else. And we will respect your own publication timelines. So we will try to organize the challenges in such a way that they don't interfere with the other purposes that you have for these genomes. The only requirement is that they're not public by the time that we run the challenges. And so sequencing, of course, we would do short-read sequencing again, but also long-read sequencing with the latest technology from Oxford Nanopore. Maybe we should also do PacBio data like we did in CAMI2. This is up for discussion. The question is, how deep should we sequence per sample? How many gigabytes pairs of sequences should we generate? Currently, we could do it like last time. I think we did 2 gigabytes pairs of sequence, but we could also do 5 gigabytes pairs for the more complex samples. So we just get a bit deeper coverage there. In terms of the methods, focus would be again on the metagenomics, on the common tool categories. Of course, if there's more important things we should focus on, we're very interested to hear about that. And then the human metatranscriptomic challenge, this is currently still in the design phase. So it would be nice that we use the same body size as the metagenomics data and the same sample numbers. So we always have a metagenome and a metatranscriptome sample for the analysis, because this is typically what people also use oftentimes when they do metatranscriptomics. And so in creating those data, we had to extend our meta-omics simulation software concept. And I had a student in the lab, Joyce Funk, who has been working with Adrian Fritz on this over the last couple of years. And so, for example, one thing that has to be modeled there is the abundance distribution of gene expression. And this is added to the abundance distribution for the different taxa in the community to then create overall gene abundances. So they are a combination of taxon abundances times the gene abundance resulting from expression. So how is a metatranscriptome data assessed? So mapping to the gold standard genomes, we know the underlying abundances of the genes that are being simulated. And then when we are creating sequence data for people to work with, we would also again do short read pattern Illumina, potentially long read sequencing, you have to tell us. And the question is also which sequencing depth would be common here. So these are the details we still have to work out. So here's just a brief overview of this new version of the CAMI-Sim software, where in this dark blue box is the innovative metatranscriptomic part. So in addition to reference genomes going in, now we also need gene annotation files. And then, so these are used to create gene abundance distributions. And then they are needed in addition to the genome distributions to create effective gene expression values and then simulate reads for these particular features that have gene expression, such as coding sequences. And then outputs are, in addition to genome distributions, we have the gene distributions, we have the gene expression data, and a lot of mapping files. And so there will be just additional data that's being generated and allowing this joint analysis. And so what are the method categories that we have to assess? So one question what people want to typically do is they do reference-based assembly. So they are mapping the reads to a reference genome to reconstruct the transcripts. And so we could assess how well does this work for different tools. Then oftentimes you do expression level estimates, and then you do differential expression experiments. And so these kind of things, of course, could also be assessed with the gold standard. How accurately do tools assess differential expression or just raw expression levels? So here we need the input by the experts, and also how should we assess it. So with that, I really want to close. I'm really looking forward to hearing thoughts that you might have on all of these different aspects. So just to briefly give an acknowledgement to all the contributors to CAMI. So as you can see here, it's a huge effort. We had in the last round of challenges more than 100 contributors from 77 institutions. And I especially have to thank Fernando Maya and Adrian Fritz and Jilu Deng from my lab, who worked for years, very extensively on this project, but also all the collaborators who contributed by implementing metrics, by submitting tool results, by providing data, by providing suggestions of how the challenges should be run, and by evaluating the results. It was just a really massive effort. And if you want to get involved and you want to know more about how your tool does or how a certain tool category does, then please do. We do CAMI for you. You can follow us on X, or if you have, and just get in touch if you have something like methods to benchmark, if you have ideas about a new challenge or data to contribute. So we're happy to hear from you. And now I'm looking forward to your questions. Many thanks.
Thank you. Thanks a lot. Any questions? So maybe I can start. Thanks a lot, Alice. Great talk. And I was wondering, in terms of like bringing real. Huh? Here in the stage. So you mean for camera? Yeah. Oh yeah. Oh yeah. Okay. Okay. I see. Where? Yeah. Okay. Yeah. Again, thanks a lot, Alice. So in terms of the data, do you have any plans to bring like real experimental data in addition to simulation data? And if so, how many samples, and like what is your vision to kind of balance between simulation data, which is obviously easy and like free of charge to generate, versus experimental data, which is like most substantial effort is needed?
Yeah, that's an interesting question. Can you see me and hear me well? Does that work well?
Yeah, we can see you well and hear you well.
Okay, perfect. Yeah, I'm so sad I can't be there. But yeah, it's great at least this way. So we had in the CAMI2, we had some real data, which was this clinical pathogen detection challenge. And in the future, we did think about that. So with the real data, if you sequence mock communities, one problem is what I was told is that you just can't get so complex. So the number of different genomes you can put together and actually be accurate about it, it's just limited to something like around 50 at most. And so we hadn't really with that gone up to the complexity we wanted to have in some parts, but I absolutely think it would be exciting to include if we have partners who would be interested in really helping in generating such samples.
Yeah. Thank you. And indeed, you're right. Like I think that's one limitation of the mock communities. You can do quite few microbial species. Any other questions?
Hello, I have a quick question. So about the data, are you going to include eukaryotes like fungi, especially in the clinical data, and also viruses?
Yes, absolutely. So we do want to include in some of the past data sets from CAMI. So the second round of challenges, there's already some fungal genomes included, and in most of them also some viral sequences. So yes, absolutely.
Yeah.
Hi, great talk. I had a question with how these tools update and version numbers, etc. Is there any way to get like almost like a live result where people can say like, yeah, I updated my tool. I think it performs better on these data sets, and do that in like a way instead of just like having to wait for like a large paper to be published to show that?
Yes, yes, that's an important thing. So we're currently working on a benchmarking platform. People will be able to submit their tool results and immediately get some feedback. So this just facilitates benchmarking, hopefully. But that's apart from the challenges. So with the challenges, because we don't reveal the ground truth until, because otherwise you could just continue to submit until you get your best result. So it's like two different things. But with this benchmarking platform, you will be able to run your tool and assess it on from past challenges where the ground truths are already available.
Thank you.